name is Pieter Koopman and I'm a bioinformatician at the Radboud University Medical Center in Nijmegen, the Netherlands. And I'll be telling you today a bit about our regional surveillance network. We've been sequencing ESBL and carbapenemase producing Klebsiella pneumoniae for over 2 years now. And these samples were taken from hospitals, general practitioners and care homes in our region. They were sent to the Radboud UMC for sequencing. And after sequencing, these samples were sent to the online platform 1928 Diagnostics. This platform was used for cluster analysis and also for databasing. Additionally, more bioinformatic analysis was performed in-house using the tool Collaborate. And with this, we hope to gain more insight in resistance and virulence, for example. Finally, epidemiological data was extracted from electronic health records to infer the likeliness of a transmission event. If we then look at our results, we have sequenced up till now 240 samples from 225 patients. If we look at figure 1, we can see that we have found so far 24 genetic clusters, and clusters are defined as less than 15 alleles difference. And if we look, we see a nice spread of sequencing type. However, we see a lot of sequence type 15 and sequence type 307, where there's diversity in 307, but there's not in 15. So that could be very interesting that there's a big cluster. However, we should note that this cluster spans a lot of time and a lot of institutes. So it's possible that this is just a rapidly expanding clone that is very prevalent in our environment. So we need to further look into this cluster to determine what's going on. Beyond that, we did additional bioinformatic analysis with Collaborate. And this analysis shows that the phenotype and the genotype are in concordance almost 99% of the time. So we see the same predicted resistance genotypically as phenotypically. If we then look at figure 2, we can actually see the resistance score plotted against the virulence score. And in this column, you can see that most of our samples actually have a resistance score of 1, which means that this is an ESBL phenotype and not a carbapenemase producing phenotype. And if we then look to this top 2 rows of this figure, we see especially here that if there's a high virulence score in Collaborate, we actually also see that there's a carbapenemase producing phenotype. And this is interesting to see why these potentially hypervirulent Klebsiella pneumoniae are also highly resistant. So we need to investigate this further in our data. And if we then quickly look at figure 3, we can actually see all the resistance genes over here. And we see quite a diverse population. However, there's an exception for CTXM15, which is very prevalent in our data. And that's also interesting to note. So to wrap up, we have managed to sequence in our regional network. We've been able to detect genomic clusters. However, it's also possible, importantly, to disprove potential epidemiological clusters. But to really do this, we need to also include plasmid analysis in the future. If we then look at challenges that we've faced up until now, it's data sharing of the potentially sensitive patient data, and also that we need to use epidemiological data to really see if our genetic clusters actually mean something or not. So to further improve in the future, we could include sequencing sharing platforms on a national basis to include more data from other regions. We could use more bioinformatics analyses to really dive into our data to find risk factors, but maybe also more potential clusters. And one of these examples is, again, plasmid research. And, very importantly, we need to assess the cost effectiveness of such an approach as this network to really see if we can have a positive impact on healthcare while also saving costs elsewhere. To conclude, we really need to keep performing this network to get more data, to look at all the figures, and then hopefully we can find out what the long-term benefits are of such a network. Thank you for listening, and thank you to all the institutes involved and all the people involved for this work.